not supported. It is what is being supported are different types of networking and communication activities and dissemination. So of course, scientific publications would be part of those dissemination activities. But what is especially supported are many activities targeted towards young scientists. So there will be plenty of opportunities for young scientists, new PhDs, postdocs, but also PhD candidates and even master students. So I will make sure to cover some of those, and I think some of those will ring a bell and be interesting for you, because just consider this these capacity building objectives. Basically, one of our ideas is to have whole new generation of scientists who will be pushing this field further on. So when talking about these actions, we usually put pictures of animals, but I have pictures of people. So you will be able to see some people who are, though this is just only a few people, of course, from the action, who hold leadership positions, and I'm putting them here mainly to know our contact points. But keep in mind that many other people are contributing to the action. We are just coordinating activities. So our action chair is Professor KRUPA Comprehensive from the University of Bologna. And our vice chair is Professor Thomas Norton from KU Leuven. So having two of them together is the reason why we have also precision livestock farming as an important part of our entire action proposal. Right now we have management committee members from 35 countries. And we have 360 members. So at the moment when the cost action was approved for funding, we had a bit more than 150 people, if I recall correctly. So during our first year, the number of participants doubled, and this is something that we are especially proud of. They're coming from 50 countries, but the management committee members are from European countries only. So I would like to emphasize that, of course, the majority of people comes from Europe, but also many countries globally outside Europe are also present in our action. We are pretty proud of our good gender balance, and also I would like to emphasize that 186 of our researchers are young researchers and innovators, which according to cost rules means anyone who is younger than 40 years of age. And 136 are coming from inclusiveness target countries. Those are the countries that are classified according to cost as less research intensive countries. And those would be parts of Central and Eastern Europe and Southeastern Europe, or Balkans, if you will. Again, as I said, very inclusive and really open to participation. It won't be surprising that the majority of our working group members are from Italy, because this is where the action comes from. But still, other countries are pretty well represented. So I would like to say that Czech Republic is actually among the top countries that are being present. So you can try this more and go around on this list. So yeah, we have very good diversity and distribution, and yeah, as you see, really a lot of different countries that are present. So this is September 1st. It keeps changing all the time, but it's pretty up to date. These are our working groups. The first three are, how to say, like core science groups. Working group 5 is about impact, and working group 4 is about impact, and 5 is about communication. And just to emphasize here, grant awarding coordinators, those are the colleagues who are handling all the applications for networking opportunities that are open throughout the action, which of course I will discuss in a bit more detail in a second. These are the working group members. Obviously, the core science groups are more popular. It makes sense because the majority of participants are scientists, researchers. But also we have the participants from industry, farmer organizations, and NGOs, or even some ministry representatives, and they are also more interested in working groups 4 and 5. But as you see, obviously, huge majority are coming from either higher education institutions or research institutes. Yeah, so these are our networking tools. Before I move to descriptions of individual working groups in a bit more detail. Our annual meetings are obviously held once a year, usually sometime around June. This year's annual meeting was held in Poland, not surprising. And we're usually trying to connect it to some other event. So whether there is a conference before or something like that, so just, to avoid excessive traveling, it's not necessary. And in these meetings, all management committee members are participating, but also always other scientists are invited. So for example, if your group has interesting research output, which you would like to share with us, you can apply and participate as, let's say, conference presenter. Or if we talk about young scientists, or we always have some guests from outside the Europe. For example, this year, one of our guest speakers was from the Iowa State University and so on. Then talking about young scientists, you see training schools here. They're also organized once a year and always with a different topic, always related to one of our working groups. This year's training school took place in Belgium, and it was dealing with phenotyping technology. So the idea behind the training school was to learn how to efficiently collect data about phenotyping technologies and infrastructure, and also how to process and visualize the phenotyping data itself. So it was a combination of research and data analysis, let's say like that. It took place over 4 days, and according to the feedback of participants, was really excellent experience for them. Then we also have short-term scientific missions. So none of these are strictly limited to certain career stage, just to keep that in mind. We prefer having, of course, young scientists, but being a postdoc or being a researcher obviously does not disqualify anyone from applying for these. So short-term scientific missions, as the name suggests, really are short, usually a few weeks, up to 3 months. Stay at another institution, which is funded by the cross-section and which will result in certain outputs, not necessarily data collection or experimentation, but also can be, for example, extended collaboration to finally finalize that paper you have been talking about but never sits together and finally finish it or something like that. And connected to this are IPC grants, or inclusive in the target country grants for conference participation. And the idea here is to give an opportunity for scientists, especially from the IPC countries, less research-intensive countries, to participate at the top-level conferences, which normally would be, well, too expensive for their institutions to afford. And the best possible flow here is to do scientific mission and then present the results of that mission in a conference. Again, not mandatory, but this is one idea of how it could be organized. Apart from that, we are also attending other conferences, and this is also something that is supported as one of our networking tools. Other than that, there are networking tools that we are developing on our own. So we have established our Young Researcher and Innovator Committee. We have 2 representatives here also attending today's meeting. And it is also working a lot towards these networking tools. So they are currently designing our outputs like newsletter, job search service. They are also discussing the best way to organize our own stakeholder conference, which we plan to have during the next year. These are some of, say, their activities. So let me quickly take you through the working groups. I won't take too much of your time. So the first working group is dealing with phenotyping technologies, and you will be able to hear one of those presentations today after my presentation, I think, right? And the idea behind this working group is to make very inclusive and encompassing map and repository of phenotyping technologies that are currently in use in Europe in different species, and also a map of R&D infrastructure in Europe. And this is in fact something that we have prepared. So our data is ready, and we are currently developing an interactive map, which will be available on our website. So you'll be able to go for, let's say, country or city or specific infrastructure. It will be pinned on the map, and then you'll be able to see the PR, the technology, the species, and get in contact with them, obviously, if you see the collaboration option with them. So this is something like not only having it all together, but also a very, very strong networking tool in our opinion. So these are the planned deliverables. Being working group one, obviously they are now under pressure to finish everything on time because their deliverables are the first ones to be produced. Then something more for molecular biologists, genome to phenome integration. Here we are talking, as I mentioned, about different techniques for obtaining molecular phenotypes, and also about ways of translationing those genotypes, complex genotypes into phenotypes and using them in Phenomics. Also very important thing here is a roadmap, which we are planning to develop to pinpoint the gaps and research needs in the field, because phenotyping is hard, and this is even more complex and underexplored in our opinion. So these are some deliverables, mainly dealing with reports and reviews, and the green ones I will address a bit later. Those are shared by different working groups. Working group 3 is for computer scientists mainly, and it is dealing with data formats. So how to report data, how to store data, how to unify the databases to make them as inclusive of different users as possible, but also to discuss the cyber infrastructure and capabilities that will be necessary to meet the needs of phenotyping and animal phenomics in the 21st century. So they are currently already working on different computational models and methods that are in use in Europe for this purpose, but are also moving towards other reports and proposition and opinion papers that they are planning to produce in the second part of our action. Then related to impact, what we wanted to have as part of working group 4 is heavy talks with the stakeholders. This will be absolutely necessary for us, because in order to realize what will be the economic impact of this action, we have to know in detail what is the current landscape in Europe. And when we say Europe, we talk about many countries, and obviously, as phenotypes are heterogeneous, the economic perception of the livestock industry in different European countries is also heterogeneous, which is why we are starting to explore more local or regional communities. And luckily, we have the researchers like our host today who are helping us get towards the people in different countries and see their not only the numbers but also the perceptions in order to really try to quantify and understand the societal impact and also consumer and farmer opinions about how and whether they see phenotyping important at the first place. So we have to explain to them why it actually is important and useful for them. These deliverables will be produced towards the end of an action, because we first need some specific outputs in order to go out and try to make people believe us that we are doing something important. And the fifth working group, as I said, engagement and communication. So within action communication through all those networking tools that I mentioned before, but also communication to the scientific community through our publications that we are planning, different review papers or white paper, and also something that we already started thinking about, something that we hope will be able to fulfill this ambitious deliverable of generating a textbook on Phenomics that will be inclusive of all topics I have been talking about. And outside of that, we also have our communication towards more general public, let's say, through classical communication channels such as our website and social media profiles, which I will show you now. So this is our website, which I will invite you to and see what is going on. So you have description of our action and so on. And then also under this tab open calls, you will be able to see all currently open calls for these networking activities. As I said, although cost by itself is not supporting research activities as the main outputs, STSMs do support research activities for young researchers. So those like smaller funds are available even for this part, or when talking about presenting your research at conferences, this is also fully eligible to be funded. Apart from that, we have our event announcements. So right now we won't have that many events because it's winter time and everything else, but from the spring, again, you can follow us and see what we are planning to do. These are just some of the events that I talked about. This was our annual meeting, so you can see all the people, and we really had a good time in Bologna. Then this is our training school. I would like to emphasize that our colleague Garrison Fleming from EU-LI-PHE organized it and really had an amazing group of lecturers and trainers, and the students, I believe, really benefited a lot from this training school. And this is our Young Researcher and Innovator Committee meeting, which happened 2 weeks ago in Florence. Whereby we discussed the deliverables of the working group 1 that I showed you that are due soon, and also developed some communication strategies. So again, if you would like to join us, we have a few free spots, so feel free to apply. You have Patricia and Stefan there, and they will be glad to talk to you further about this. Yeah, so if you want to join us, you can do it through our website, but also you can go to the official Cost Association website. And this is our code CA112. And there you will have the button to apply. The only thing you need is your active eCost profile. And our action chair is very up to date. He's always revising the applications and so on. So it's really not that time consuming. It can all be done in like 10 minutes. And we also have our social media profiles on all social media. So you can also follow us on Instagram, Facebook, LinkedIn, or X, and see what is going on regarding our action. And thank you very much for your attention. So if you have any questions, I can answer, or we can discuss during the coffee break. I'm here.
So thank you, Adna. It was a great lecture about UI. And I would like to ask you about questions. Do you have any questions? I think it's very important for Czech agriculture to know that there are a lot of possibilities of Phenomics and Phenomics, and a lot of people are working on these problems now. So really thanks for the introduction of the new topic for us. You work with behavior. Can you summarize how much you are interested in behavior?
Yeah, so regarding the opportunity that we have, behavior analysis is mainly done through image and video analysis. So we have research groups in Switzerland that is their main partner in this regard. That is doing peak behavior analysis.
Yeah, so Sebastian Guma probably.
Yeah, he
Yes,
is one.
because he's a former colleague.
So they actually also had one that was a she had one SDSM opportunity recently, so probably she will have something else in the next year. So yeah, we can follow that. Or if you're interested, maybe the two of you can get in touch and do some exchanges there.
So, I have a question in that. Thank you very much, Adna.
Thank you for inviting me
And we'll continue with Amelia. And I stop sharing now. Hello, Amelia. Hello,
Hello.
Helen.
Hello, do
Can
you hear
please
me?
start?
Hello.
Hello, so I started to share my screen. I don't know if you see it correctly.
Yes, it's
Thank
okay.
you.
Okay. Okay, I do not hear you anymore, so everything okay from your side?
It's okay. now.
Okay, perfect. So thank you very much. So I will start now. So I'm Amélie VANLIERDE from Belgium. I'm working at the CRAW, so it's the research center. And I will do this presentation with Hélène, which is also here, and she is from the University of Liège-en Agrobiotech. And so this presentation is a collaboration between our teams. We are working together on this topic from since 2011. And there are also some slides in collaboration with the European Milk Recording Organization. So we are talking about here the prediction of methane emission from cattle using the milk mid-infrared spectra, and more especially about the importance of the robustness of the model to be sure that the model has good performances, even more if we cross the border when we apply this kind of model. So first of all, here is a representation of a milk mid-infrared spectra. So as you can see, there are some absorbance in function of the wavelength, and the wavelength, it's kind of light we will put through the milk, and for each wavelength we will have a part of the light which will be absorbed by the milk. And so thanks to this information, we will have a representation of the composition of the milk, so the chemical bonds, and all those chemical bonds together permits to make models to predict, for example, here, the fat composition, the protein, lactose, and so on. So those are main components of the milk, and so the spectra is a representation of the milk composition. But here we are trying to predict methane emissions, so it's something completely out of the milk composition, so it's an indirect prediction. And it's particularly interesting to use the milk mid-infrared spectra because this spectra is already obtained in routine. It's something which is non-expensive, which can be obtained fast, quickly. And so it's a good definition of a proxy. And using milk composition, so milk with infrared spectra, to predict methane is relevant as there is a common origin, the ruminal fermentation, which will impact the methane emission and will also impact the milk composition. this is why we make a link between meal composition and methane emission. So to have a robust model, it was the second part of the title of this presentation. What is a robust model? It means that it's something that that will provide good prediction under various conditions. So it's like if you compare a very, very fast car, which will be very performant only on the road, but when you go out of the road, it's not performant anymore. It's not something robust. So here we want to have a model where we can apply everywhere. It's like very performant on each type of condition. This will permit to avoid extrapolation and overfitting. This paper from my colleague Clément is very interesting to read for this aspect. So I invite you to have a look on this paper. And so to develop the model to predict methane emission, the point was to first create the model and collect data. So it means methane measurements and the corresponding milk mid-infrared spectra, taking care to consider of a maximum of variability, existing variability. The very first model which has been developed, so this paper is from 2012, it was the demonstration that it was relevant to consider milk mid-infrared spectra as a proxy to estimate methane. And as you can see, the statistics are very interesting with the R-square of cross-validation of 0.72 and standard error of cross-validation of 96 grams per day. It was very interesting for a very first model. But it's not robust at all. Indeed, it's only built on 11 cows. Okay, 3 types of diets, but very few animals and very few conditions. So it's a first step, but very not robust. And so this is why we wanted to build an international model to predict methane from milk mid-infrared spectra. And the fact that it's international, we can use it in a maximum of situation. And for that, we need to include a lot of variabilities in plural in the calibration set, meaning variability at zootechnical level, so the breeds, the diets, physiological stage, and so on, the CH4 range, spectral level, etc. So for the moment, the model that we are using and sharing with some collaborators is built on data sets including more or less 1,000 data, a little bit more. And it's the result of a collaboration between different teams within different projects that are represented here from the main ones. We had access to reference data obtained with the SF6 technique, as you can see here from Belgium and Ireland. And also data with obtained with respiration chambers for methane measurement, I mean, from those countries. Of course, we always had the spectral information in parallel. And as you can see, the average of the emissions are also different depending on the data set considered. And it's also due to the fact that different diets were tested, different conditions and so on. So it doesn't mean that in Belgium, for example, the emissions are higher in general. It's because we wanted to have variability. So we tested different types of diet to have high value, low value, and so on. So here as sources of variability, I mentioned the breed. So you can see here we have Holstein, Jersey, Brown Swiss, Red Holsteins, cows and crossbreeds. But we have a large majority of Holstein cows. The variability is also about the lactation stage. So here, once again, you can see we have data at different level, but the main data are in the middle of the lactation because it's always easier to have data at this period than, for example, at the very beginning or at the very end of the lactation where animals are not, it's not common to put just in a respiration chamber, for example. And here is a representation of the spectral variability that we have in the model. So here is the colors represent different countries. And here is the exact same graph. So the principal component, three first main components. So the exact same graph than before, but depending in function of the breed. So we can, for example, see here that the Jersey cows at spectral level are clearly a little bit different from the other. It's expected. As for example, for those cows, it is known that the fat level is higher than for Holstein cows. This is why it's important to include them in the model, because if we want to apply, for example, a model built only on Holstein cows, and we want to apply it on Jersey cows, we can clearly see already at this stage that the spectral information is different. So we will try to have a prediction from a different spectra, and so it will be an extrapolation. This is why the first step when we apply a model is always to see, okay, is the spectra covered by the variability of the model before applying it and obtain a prediction value. Here is a representation of the measured data in function of the predicted data from this reference dataset. So it's clearly the datasets that represent the prediction model. So we can see that we have a clear relationship between the measured and predicted value. We have a little bit more than 1,000 data and the R-square of cross-validation. So the error of prediction is around 57 grams per day when we use the model in those conditions. The data are coming from different countries, as mentioned before, and based on reference value acquired with SF6 technique and respiration chamber. So it's a very evolutive model. It's something that I really want you to understand. So from the very beginning, we had different steps. Whoops, sorry. And I will not detail every step because it's a long story, but to be short, we also include the lactation stage information in the model because we really saw that it was important because the milk composition is impacted in function of the lactation stage. For example, at the very beginning of the lactation, the cows are in negative energy balance, and this will also impact the source of fatty acids in the milk, and this will be a bias for the link that we make between the milk composition and the methane emission. So we included this information in the model to correct this bias. Then we tried if it was okay to merge data acquired with SF6 and the respiration chamber. That was a very important step because the reference value is very important. And of course, if you use reference value which are not so reliable, then the model will be even less reliable and so on. So we tested this step. We also try to consider additional zootechnical informations in the model to improve the model, such as milk yield or the breed. And also, of course, increasing the reference dataset to be sure to cover a maximum of situation and so have a model more and more robust. So those are more or less all the publication about this topic and the different steps that we followed. And it's important to have in mind that we will continue to make this model evolving in the future. Of course, if you have access to new reference data with the idea of this evolving model, contact us because it's always interesting to challenge the model and to also improve the model with the implementation of new datasets in the reference datasets and to have a more robust model. It's a win-win situation because in that way, the model will be improved and also the person who give access to reference dataset to improve the model will have access to the model back and be sure that the model will be relevant in their situation. So now I will give the floor to Hélène for the next slides.
Okay, thank you, Amélie. So, good morning, everyone. Thank you to have invited us to present you the methane. So now I would like to discuss with you concretely how we can use the methane predictions. So, to have access to the model, so as Amélie said, it's a win-win situation. This is really something that is very important for us. You have seen that we need to have a lot of data. We need a lot of phenotypes, meaning reference phenotypes, obtained in different conditions. We believe that we can create only one model. We don't need to have a lot of model, but it's always better to have a general model, meaning not a model developed for a particular region, but globally on a European scale or more. And this is exactly what we try to do with the development of this model. Why is a win-win situation? Is just because we, as Amélie said, you give reference data and back you have the model. So the first option is the research collaboration. And so in this case, you are the data provider. So it means that you give us the reference value, but it's very important to notice that those values are not shared between partners. We are, as developer, the only person who has access to the full database. So I think that this is really important to notice. But after that, what we do, we include your data into the model, then we have a new model, and finally the model is shared between all partners. So it means that even if you have given data 3 years later before, if we have a new partners now, it means that we will have a new models. The models also will be shared with the new partners, but also the older one. So it means that it's also an insurance to have all the time the best model. But an important aspect for that, of course, is to have standardized procedures for the sharing of the data. It means that we need to follow right protocol, of course, to collect the reference data, but also reference protocol to collect the spectral data. And this aspect is often missing just because for the people, they think that as it's coming from the MIC lab, directly the data is of quality. But this is not really true. The spectral signal is never corrected. Just to give you a comparison, when you have the measurements of the fat and protein done in the MIC lab, you have a verification of the quantification using sample with known content of fat and protein. And thanks to this analysis that is called ring test, you are able to correct the value given by the instruments for a certain bias and slope. But this is not possible to do for the spectral data until now because it's only done for protein, for the prediction of protein or the prediction of fat, but not directly on the raw spectral data. So recently we have developed a model. I will explain that to you with more detail just after, but keep in your mind that it's very important to have standardized spectral data in order to have a high quality training set to build the model. The second option is the European milk recording. It means that in this case, you have not a reference value, but you would like to have the prediction coming from the equations. In this case, it's possible to ask to the European milk recording to have some predictions. But the EMR, is also another important aspect, is also the creation of the standardization of the spectral data. So maybe we can go to the next slide. Yeah. So what is the European Milk Recording? It's an association of 13 different members, which are milk recording organizations coming from 7 different countries. We have collected data for more than 5 million cows, and we cover 86 different infrared, mid-infrared spectrometer in 25 different partners labs. So, and this is very important in order to try the standardization process, because we share all data together in order to develop new tools. And of course, again, even if it's not here really the development of an equation, but maybe the application of an equation, is very important to have a common database and also standardized procedures. So it's, as I said, we need to standardize the spectral data. So this work was done by Clément Grelet mainly in the Walloon Research Institute. And so what we need to, why we need that is really to put the data together. But sometimes people think that we don't need to make this standardization if you use the same spectrometer. But this is not true because the signals can change through the time due to for instance, the cleaning of the instruments. But this standardization, which is a little bit different than the standardization proposed by the manufacturer, it's the fact that the standardization proposed here by EMR allow you also to combine data coming from different brands of spectrometer. For instance, you can have some lab working with Bentley machine, another lab with Foss machine, and the standardization proposed here allow to have a common structure for both instrument and at the end having only one common database, spectral database. So how it works is more or less like what is done for the usual ring test for fat and protein. So the idea is to send samples to different lab, meal sample, I mean, and those samples are analyzed by the lab, and then the spectral data are given back in the system. And you can see here on the right, the blue point represent the raw spectral data. It means that this is the results of the analysis of the set of samples sent to all labs. And this is the results for all labs. You can see that we have more or less a certain spectral variability. Again, it's a representation using principal components because the spectral data contain around 1000 points. So I cannot show you that. So a good way to represent the spectral data is using the principal component. So the blue point is the raw values. But we know that we need to have exactly the same results because all labs analyze exactly the same milk samples. And so then using mathematical conversion equations, it's possible to standardize those samples. And the red point represent the masters. We have decided that some machines, which are particularly well checked, are the master. It means that they are the goal, they are the results that we need to obtain for the analysis of those samples. And you can see now the red points represent the results of the blue points when you have applied the standardization coefficients. So you can see that you can see that we decrease a lot the noise around the spectral signal. And this process is done more or less every month in each participating labs. So when you have standardized spectra, then you are able to apply more or less any equations. But as Amélie said, in order to avoid extrapolation, it's very important to know the context in which equations were developed. So it means that if in your country you have very specific feeding or specific breed, it's really important to ask first if the equations are developed under your conditions, because if it's not the case, it's really not a good idea to use an equation. But here, through EMR, so meaning for European country, we have developed, of course, model for protein, lactose, urea. This is well known. But we have also model for fatty acids and casein. Those models are very specific because they are directly related to the milk compositions. But through the time, we have seen also that the milk spectral data can be a good mirror of the cow health. It means that we have a direct link between the milk composition and the metabolic status of the cows, for instance. It's why now we have developed a series of indirect models like the quantification of acetone, BHB, citrates in milk. We have also BHB, NIFA, for instance. We have also developed, and this is an indirect model, equation for minerals, because minerals has no direct fingerprint in the spectral data, but we can be able to measure those components thanks to the link they are with more major component in the milk. Then also we have seen that it was possible to develop equations to assess the milk properties. It means that is the ability of the milk to be transformed mainly into cheese. So we have also developed equation for that. And we have also equation, of course, for methane, as Amélie present the results, but also for the energy balance and nitrogen efficiency. So again, for all of the equation that I have already discussed with you, if you bring reference value, you can have back the model, because again, for all of those equations, it's a win-win situation. So if you are interested in, you can just contact Amélie or me, and we can also make this collaboration alive. But if you have no reference value and you want to make these predictions, you can use the EMR platforms, and 3 different packages are proposed. Packages A with the prediction of methane and fatty acids, packages B for BHB, acetone and citrate in milk, and package C for minerals and lactoferrin. So now, more concretely, why, we need to have the methane predictions, especially using infrared analysis. I think that the main important point is that you are able to have a lot of reference value. We know that having methane phenotypes is a very hard work because it's not very easy to develop that at a large scale. But using the infrared spectrometer, it's possible to do. So based on that, you can make some advice to breeders. This is already done in France through, for instance, the projet Laitage. And you have also another project by Alliance. And the idea is really to give feeding advice and feeding strategies. Another possibility is to conduct large-scale studies to study the phenotypic variability of methane, meaning observing the difference, of course, between feedings, but also between herds, between seasons, region, breed. And also, it's a very important aspect because maybe you can discover that some management practices can be beneficial to reduce the methane emission. If you can do that at the phenotypic scale, of course, you can do it at the genetic sites. So it means that when you have a lot of data, you are able to estimate a breeding value for specific cows. And so you can conduct usual quantitative genetic evaluations for cows and bulls. So the next step, when you have breeding values for bulls, you are able to conduct genotype studies. So this is the next slides. And so we will have soon genomic evaluations for residual CH4 in methane is expected around the end of this year. And in Wallonia, what is very interesting is that we have a lot of bulls coming from different countries. And so this is very nice because maybe we can have some bulls that are already used in other countries and shared the results that we have. So now, of course, it's not perfect. And so it's very important for us also to provide a nuance because what is the limitations for the use of these predictions of methane coming from the spectrometer? First, the strength. We have already said that the acquisition of spectral data can be done routinely through the milk recording. In this case, you have data more or less between 4 and 3 weeks at individual scale, but only for herds participating to the milk recording. But you can have also spectral data through the analysis of the milk use for the milk payment. And so in this case, you can have spectral data, but now represented the herds, but every 1 to 3 days. So the frequency of data acquisition is really, really high. So this is really, for me, the most positive aspect of the MIR. It's also a very fast analysis. It's cost-effective. Is very few cents per analysis. Thanks to our techniques, you know the prediction error. So this is also a very important thing because we are very transparent about the obtained results. It's scalable. It's a scalable method, and it's really close to the physiology because, as Amélie said, we have a link between the meal composition and the rumen activities, and it's based on that. We can make this equation. So we have really a link with the physiology. But of course, we have limitation. It means that if we want to avoid any extrapolations, the equations can only be applied under context that is known by the equations. It's why we continue to collect more and more data to improve the model. We need to have a variability in terms of spectral variability, of course, so different meal composition. We need to cover different diets, different breeds, maybe different meteorological conditions because the heat stress can also impact the meal composition and also the methane eructation. And another limitation is the diet additive. Of course, if you change the rumen activities and if you broke the link between the rumen activities and the meal composition, of course, our equations cannot be relevant because in this case, the meal composition will not be a mirror of the methane eructed by the dairy cows. But at another side, but we still need to prove that, but I'm more or less convinced about it. Uh, even if you broke this link, of course, the estimated methane will not be the true one because you have broken the link between the eructation of the methane and the meal composition. But I'm convinced that the prediction that is done using the spectral data represent the biological ability of the animals to make the prediction to eruct methane. And this can be very useful, especially for genetic studies. So I think that even if you use diet additive, I think that the prediction of methane through the mid-infrared spectra is still relevant for genetic and genomic studies. Another limitation is that we need spectral standardizations. We need to have a common format for the spectral data. And another limitation is that this estimation of the methane predictions can be only provided if you have lactating dairy cows. So what about the first parts of the life of the cows, and also what about the eructation of methane during the dry-off periods? We need to have other techniques. Amélie developed other techniques using infrared based on the phases, but in the case of using mid-infrared, it's not possible. We need only to do it on lactating cows. But I think that it's still a good point because it's the main parts of the life of the cows. So now the take-home message, what we would like to say to you, and the main message is that we are transparency about the methodology, about the data set, about the data provider, about the prediction error. So I think that this is very important. There is a very huge potential of applications at individual scale, but also Earth scale, because we have different frequency of data acquisitions and we have a large variability. And I'm pretty sure that we have also some sources of variation that we don't know. It's very important to mention that it's, as you have seen, the first model seems to be very interesting because the statistics were very good, but at the end we have seen that the model was not enough robust. So please, it's not a good point to focus only on the matrix, the statistical matrix, I mean. It's very important to see the behavior on your model on the field, and to see if you have the expected behavior, for instance, based on the stage of lactations, the season, the breed, etc., based on what we know from the literature, and also reference measurements. It's very important to limit the extrapolation. You cannot use the equation if the equation was not built for your context. So need to cover local variability. Importance of collaboration. We are very convinced about the win-win situation. It's already promoted through the Extramere project. So if you don't know this project, it's a project managed by the International Dairy Federation and the International Committee for Animal Recording, so IDF and ICAR. And in this project, we try to show the importance to collaborate in order to obtain and very robust model developed for a large variability of context. It's also a good way to decrease the cost in the development of the equations. And I think that this is really important, especially when the acquisition of the phenotypes is expensive or rare. And finally, we need to standardize the procedures for the methane acquisition and the spectral data in order to be sure to merge data and to have at the end a high quality for the training set used to develop the model. Okay, so everything related to the method used to obtain the methane, because we forget that, but we can obtain methane phenotypes, reference methane phenotypes coming from different techniques. And so it's very important to know the protocol, the best protocol. And so a good article was writing by KRUPA about that, just to be sure to have high quality reference value to build your model. Another maybe short comment for you, it's very important to notice that having methane data coming from respiration chamber, SF6, sniffer or green feed, it's more or less not the same measurements, it's different measurements. We have seen that it's possible to merge together SF6 and respiration chamber because they are representative of a 24 hours of methane emissions, but it's clearly not the same for data coming from green feed and sniffer. So this is also important to keep in mind. Thank you for your interest. And so I suppose that we have questions for Amélie and me.
Oh, thank you very much. And I will ask the participants for questions. Do you have some questions on Amelia or Helen?
I will maybe just take time for the reflection of question to add that, so we mentioned that for the moment, the model shared is based on SF6 and respiration chamber. We are also working on another model based on green feed measurement. And so if you have measurement reference data with a green feed system, please do not hesitate to contact us also.
Hello. So there are no questions, I think.
Okay, so we were very clear. That's good.
If you have questions in the future, do not hesitate to contact us by email or something.
May I ask you a question? So both of you. I would like to know if the same method can be used for feed efficiency instead of methane.
Yes, there are some models which are in development for feed efficiency, but I'm not the person in charge of this. Yes, you can consider this. It's not exactly the same aspect as you need as reference value the feed efficiency, but you can do the same process. So having the reference measurement, the spectra, and then build the model. So I have in mind a team from INRAE working on this topic. So maybe it's the best to have a look on those papers.
Oh, thank you very much. So thank you very much, Amelie and Helen. And it was very nice and informative lecture. And now, so thank you very much. We will contact you, especially by emails, because we have to think about this problematics, and it was very interesting. So thank you very much.
Thank you.
Thank you. Have a good day. Bye.
Bye.
Bye-bye.
So now I would like to ask our colleague from Brno, Petr FLEISCHER, to share his presentation.
Okay. Good morning, everybody. I'm very honest to present some our experiences and results from traditional phenotyping, let's say, about health data. So the title is Voluntary Harmonized Record of Health Events on Cattle Farms in Czechia and its use. I'm from the Veterinary Research Institute, but let me first briefly describe our Czech dairy industry. We have approximately 360,000 dairy cows, and almost 2/3 of them are Holstein breed, with milk yield, average milk yield about 10,500 kilogram milk. A little bit more than 1/3 are Czech Simmental type flag fee cows, which is dual purpose breed with a little bit lower milk production, almost 8,000 kilograms. But a very important aspect in our country is the size of the dairy operations, which belongs among the biggest in the European Union. I can describe it that almost half of our cows are kept in operations with more than 500 cows. If we take the cut point 300 cows, then more than 3/4 of our cows are kept in operations with more than 300 cows. Such large size creates a little bit specific conditions, enables workers, management, specialization, and has also influence on technology used, on request for recording, and so on. Our dairy cattle, almost 20% of Holstein heifers are genotyped, and especially Holstein farm have got professional management. On the other hand, some weaknesses is to find motivated and reliable workers, but it's probably not only our situation. Regarding breeding, we can say that there is realized balanced breeding covered by Czech Holstein Association, and important is that in the last, let's say, decades and years, there is a growing emphasis on fitness traits and slowly also on antimicrobial use. Antimicrobial use has a relative close connection to health data and phenotypes. The position of the Czech Republic regarding the total antimicrobial consumption is quite good. But if you would see a chart graphic with critical important antimicrobials, our position wouldn't be so good. Despite this topic about antimicrobial use is a different topic and story, I will not speak detailed about it, but I wanted only to say that I regret that our state doesn't use the opportunity through the request regarding in accordance of European Union legislation there is possibility to request the antimicrobial usage at end users. This is not completely used in our country, and it would be large opportunity to get health data, especially at cattle, dairy cattle. Let's describe our own story. Approximately 10 years ago, our two research institutes and National Czech Moravian Breeders Company started to prepare a project where we wanted to work with health data. So the challenges were how to get the data, and I only want to note that at cattle, all animals are mandatory identified, have own unique identification. So we speak about individual animal records and data, but the type of documentation was very variable. Some farmers used still paper for health data recording, and there was approximately 20 farm management softwares, both international or from international or national companies, but as far as I know, no of them used were online, at least dominantly didn't kept the data, health data online. And some basic problems were that there were no uniform system of the names of health events or diagnoses, and there were no central database for health data. We created it and started to run it from 2018. The basic goals of our first project were simply to help farmers with mandatory treatment records, with health management on all the levels, possible levels, and from the longer time perspective to enable breeding for direct health rights. We created a web application, online tool, Diary of Diseases and Treatments, simply we call it Diary. And it's also a short description about it is also on a web page of an international organization dealing with antimicrobial usage. Our online tool, Diary of Diseases and Treatments, is a web application free for farmers, which are in the milk recording system. And as basic fundaments are that this application is connected to official national animal registration database, is based on modified ICAR Central Health Key, which is a list, basically a list of terms, diagnoses, and second most important part is daily updated database on approved medicals, because both these last points are very important for possibility of compatibility, for harmonization, because in the time before, every user were able to create his own names for health events, his own abbreviations for medicals, so it wasn't possible to be compatible. In our system, the users can't create such items. They can only choose from the list of health events and from the list of medicals. So the input of health data are two basic ways. The direct entry by farmers directly to our web application, or second important way is from third-party softwares using a web service a kind of automatic data sharing. The example of a page with a direct entry of health data, as it see the farmers, and I wanted to highlight that the blue color of items they can choose means that these blue items are important for breeding. The motivation of our farmers is to provide, to use our tool, which we hope saves time and money for them and enables them management decision with the help of lists of individual animal health history, when they should decide to treat or not to treat the cow, for example. Or they use list of animals for repeated treatment or control, or can check a list of animals which are in withdrawal periods. We created also, let's say, plenty of output statistics and graphics. For example, structure of disease and medications per different time period, months till year. They can be divided by categories of animals, calf, heifers, cows. There can be also overview of medication costs. And as I already noted, there is a systematic treatment record keeping as it is requested by our national law. In relatively new output is benchmarking, which I will show later. This is an example of some basic herd overview of health data from for example, last 4 months. But I would like to say that the outputs can be modified by farmers according their needs and questions. In this basic overview, the let's say red figures are interactive. Also, the names of groups are interactive. So there are more levels of where the farmers in the outputs can go. This is the example of Holstein herd and population benchmarking. And without going to very details, I can only briefly show that this is an example of a herd which is well producing over average, because red lines are the results of the own herd compared with population median. For these columns are median in the middle of the graphic. So you can see that this herd has got quite acceptable somatic cell count, average somatic cell count of the herd and of the older cows, but they struggle with clinical mastitis, especially at primiparous cows. So, especially in the field of breeding, our Czech farmers have also, besides our project, quite large amount of more than 50 traits and indexes they can use for many aspects of breeding. And from the health traits, thanks to our projects, it were created and introduced breeding values and genomic breeding values for clinical mastitis, the same for food and claw disorders, and more health traits and aspects are then combined into health index, which is topic where you can maybe speak, and colleagues from Animal Institute later today, and you can ask them to do more them for this topic. So I can summary to this web application dairy that this is a powerful tool for farmers, but also vets interested in herd health management can use it, and also the farmer can allow access to other specialists working with them in, let's say, knowledge team, like a nutritionist and so on. And we succeeded to create a solid base for genetic evaluation, because our web application covers approximately 20% not only of dairy cows, but all animal categories at cattle, which means that also covers calves and heifers. And especially in the now started project, we have the goal to focus on health traits at calves and young stock. But everything is in development, and we would wish and see that we would welcome more connectivity and more data exchange, which is needed more and more. We work on to create such connectivity, especially with hoof trimmers. And only small note that I mentioned that we try to cooperate both with national and international companies who are also using their farms management softwares. We are successful in cooperation with national companies, but with international companies, it's less. It's a smaller success because larger international companies are not yet very interested in sharing the health data. But okay, I hope did some progress, and I thank you for your attention, and we'll very welcome any questions right now or through the contacts I provide here. I only wanted to ask you for direct questions right now after during this discussion, because I apologize, I'll be not present at the final general discussion in the afternoon. So thank you very much for your attention, and I'm available for questions. I will welcome them. I will stop the sharing. Okay. I can't hear the organizers.
Yes. Thank you. And thank you very much for your lecture. It was very nice. And I would like to ask participants inside for the question on about the health of cattle and so on. Do you have some questions, someone? So there is no questions. So you can contact Dr. Fleischer by email or by phone, or I can give him some your next question through the general discussion. I hope we will have in the afternoon. And thank you very much, Peter. Thank you.
You're welcome. Bye-bye.
Bye. And the next, I think it would be better to have some coffee break because at 11, I hope Chair of Action will join us through the Zoom and Professor Fontanesi give us some welcome speech or greetings, yeah, at 11. Okay. Yeah. So now we can
discuss
yes,
and prepare a lot of
enjoy
questions.
discussing and sharing ideas.
Yeah.
So,
Oh, thank you.
thank you.
All right, so thank you for inviting me to give just a late welcome to this national, regional event of the European Network on Livestock Phenomics. I really appreciate the local organization. Ludmila did a great job, and also with the collaborators in order to establish a network, a local network, and try to enlarge the European concept around the livestock phenomics. So I think it is really important to talk with the stakeholders, with the scientists in different ways. And I wish you the best for this event. Unfortunately, I could not attend. I come in Prague. I think I will be in Prague in the future, I hope. To join you and visit you in the future, but there will be many other opportunities to meet using our cost actions. And thank you very much for organizing, and I wish you a great meeting and successful meeting. Thank you. Best wishes. See you. Unfortunately, I could not stay here for the meeting because I have other duties in a few minutes. And thank you again. Bye.
Odmutujte se, prosím.
Audio, tak.
A chtěl bych to vyšerovat ještě. Tady, promiň. Tady. It is okay now? Audio, microphone, okay? Hopefully. Okay, yeah. And hopefully fine. Thank you. And my name is Emil KRUPA, and I would like to present here 3 lectures. Don't worry, so boring as found. But if you sleeping, I will wake you up. Yeah. So first is oriented on comprehensive processing and use of data obtained from recording system in livestock breeding. The next one is about genetic evolution of pigs in Czech Republic. Actually, it's about the new phenotype using in the genetic evolution. And the last one will be oriented on utilization of economic data in breeding. Everything okay? Okay. At the beginning, because I'm here as a representative of Planted Limited Company, let me briefly introduce our company. Our company is a subsidiary company of Czech and Moravian Breeders Corporation. And this Breeders Corporation, let's say this is umbrella corporation or umbrella organization for animal breeders, mainly owned by Breeders Association. And one of the main responsibilities of this corporation is official animal registration movement, also IRTAC distribution, milk recording, and this our institution or our corporation respond or let's say that's in our genetic laboratory. We analyze the DNA, mainly DNA, and perform parentage verification. And 3.5 million of samples are analyzed in our milk laboratory per year. And in close cooperation with research institutes and universities is carried out genetic evolution of the cattle. And also the organization is responsible for storage of tissue samples in cryobank. And nowadays it is in the preparation a new bovine semen sexing laboratory in our corporation. And our institute respond to the grant calls in which it actively cooperate together with research institutes and universities. Actually, we cooperate on 1 international and 4 national grants, and 1 international and 3 national grants are submitted and under evolution. Our institution, our corporation is member for Czech Republic in ICAR and ISAC. And a lot of our activities are awarded by the certificate of quality of ICAR organization. For example, milk and meat performing testing, exterior evolution, breeding value estimation, parentage verification, all DNA laboratory analysis, and so on and so on. So the Planted Limited is a national data center, and that's it. Yeah, it is a workplace. Very specialized to processing of phenotypic breeding data. especially for milk and reproduction performance testing data from exterior evolution from meat phenotypes. This is especially for Czech FLEISCHER breed. In our company, we evaluate and process and publish the breeding values. The process of estimation of breeding values are performed in our institution only for Czech Holstein population. And let's say all these phenol, genome data are processing, storage and publishing in various ways, especially as web services, web pages, data files. It's mainly oriented on the electronic way, not for papers. Yeah. And we are using a very complex information system which allows to insert new data or modify existing data only by authorized users. That's very important. The data come to Blended only by electronic way from the various sources like test day reporting or animal reproduction reporting or meat or dairy yield reporting. There are some reports from the Build laboratory, automatic reports from, let's say, from automated milking robots and so on. So there's a lot of sources, and this data also come in a different periodicity. If you understand what I can say, the system of processing have to ensure that this data remains up to date and with compatibility with other data, with other aspects. The processing of the data is very important, and on one side it's based on the, let's say, very complex logical rules to prevent, store misplaced data. A lot of these rules are very are based on let's say on logical breeding rules. And on the other hand, there are a lot of, let's say, internal or methodological rules. We are using for let's say calculated traits or derived traits, derived data. The results of the data should be on one side the validation of the data and storage in the system, or the denied storage due to some let's say mistakes, and subsequently sending the request to correct the data to the users. At the end, the validated data could be a source for genetic evolution. As I mean, we are performing genetic evolution for Holstein breed, Czech Holstein breed, and he is some traits. There's complex of traits. We are estimate breeding values for production traits, somatic cell score, linear traits. There are 25 traits. Also for fertility, longevity, calving disease, and so on, speed, milking speed, gestation length, and some clinical mastitis. The development for breeding value estimation start at 90s years by the Dr. Chermak. And lab animal model for the major traits was developed during 90s. Including the test day model was performed in 2005. In 2011, since 2011, beginning development of single-step GBLAB method. Since 2014, we publishing the first official genomic breeding values for production traits. And in 2015, we finished the in-house program for all traits in National Holstein Index. abbreviation is in Czech language. And since September 2015, we done a validation for production for Gmail interview, which was, I think, the first in the world. Next, in the 2016, we are using new programs, new software for estimation genetic parameters and breeding values for the family from the family BLAB F19. Since 2018, we are estimate genomic breeding values for milking speed. Next in the same year for linear traits, and we are performed first SSG BLAB with pseudophenotypes from breeding values from maize. In 2019, genomic breeding values for traits for production traits, somatic score, longevity was performed firstly. And in years 2020 and 22, we are incorporate the new traits, robotic milking index and gestational length. And in last year, we incorporate the new, we start incorporating new health traits to the routine national genetic evaluation. Here is a very simple, say, process chart of estimation of breeding values using by step method for Holstein breed. We start with phenotypes for exterior and stud, and from that data we estimate conventional breeding values. Then we send it to the interval, to the maize prediction base compared with breeding values from other countries. Results from the interval come and we calculate some pseudophenotypes, and these pseudophenotypes are using together with phenotypes and SNP data from laboratory to estimate the genomic breeding values by SSG method, and then the results are published to farmers. Next chart, next process is very similar, but is used for estimation breeding values using by blending method. We also start with phenotypes from exterior traits and from stud. We estimate the conventional breeding values. Then also we send it to the interval for maize prediction, and these results together with estimated conventional breeding values and SNP data from laboratory are used for estimate breeding values by blending method, and then is published to the farmers. So this is very briefly intro about our company. Our institution, and I will continue a little bit less boring about using new phenotypes in pig breeding program in Czech Republic. Nowadays, we are using a national pig breeding program for pigs, where we published I think 16 breeding values or total breeding values for silent dam breeds, for 5 breeds together. And the situation in Czech Republic is that we have only test on farms. We have no data from stations. And the tests start at the age of approximately 80 days, and these tests are managed to age of animals due to simplicity for farmers. And only one result for this data is average daily gain and limit content. But there is a big economic impact of feed traits on profit, and therefore breeders decide it will be good to incorporate some traits from feed intake in the breeding evolution, in the breeding value estimation. Therefore, since 2018 start research project which was focused on collecting a new type of data from the automated feeding boxes, feeding system, which also covers analyzes which factors affecting these traits, feeding intake traits, and will results with estimation of genetic parameters and breeding values. This is work of my colleague from Big Breeder Association, so from Research Institute CH4, and the research started one farm which was equipped by the automatic feeding boxes, and the animals were or still are feed in boxes during the whole test. But as you can see on the left chart, there are a lot of outliers in the weights, and because we need this data to be able to analyze for some the subsequent process, these data have to be edited. The main reason why are the outliers was or are when some animals stepping on the scale, for example, stepping on the scale when the animal in the feeder box. Yeah, so the editing of data was done by identifying the not logical weights, which was done by compare these traits with let's say subsequent, and this was done by using regression, linear regression on weights with that was on body weights and usually gain and something like that. And it was performed 20 times, and results after editing is shown in the right chart. And after then it was estimate genetic parameters like okay, before we analyzed three traits, the average daily feed intake, feed conversion ratio, and residual feed intake. Residual feed intake is the difference between observed average daily feed intake and expected feed intake with some expected values, which was used by the formula, by the equation, which is you can see the formula here, and result is some genetic parameters for these three traits. For average daily feed intake and feed ratio, this heritability about 35%, and for residual feed intake is higher, approximately 43%, and it was observed higher proportion of variability explained by Penn or by batch. It was approximately from 20 to 60 to 47%. It's a lot. And this is all about using the new phenotypes in genetic evolution of pigs. And the last part is oriented on using economic data in breeding process. We use this data especially for not only economic data, but this is complex of inputs covers production, reproduction, veterinary, and costs, revenues, a lot of categories. And these data are used for calculation economic weights or economic values for some production, reproduction, functional, and so some traits. And these economic values or economic weights are used or can be used for construction of selection index usually, and also can be used in breeding programs. In our institute, we developed the program package EcoWeight, which is mainly used for calculation of economic weights, and they also can be used for calculation economic or for use for economic evolution and for the system optimization, for a lot of marketing strategies. And nowadays we have let's say 4 separate programs, each for some species. We have a program for cattle. There are actually 2 programs, one for beef cattle and one for dairy cattle. And the number and the quotas represent number of traits for which economic weights are calculated. So we also have a program for sheep and goats, for pigs and for rabbits. And what is interesting, we from time to time update the programs, we incorporate new traits, which is done usually to request from the user from the countries. Yeah, now we incorporate new traits for sheep and goat program. Actually, the program is used mainly, of course, in Czech Republic, for example, Holstein Association and Pig Association use it for create selection indices. But for example, now a cattle program is used by Qantas in Switzerland. As I mentioned, from University of Given in Germany, they asked us to incorporate new traits, so they are using it now. In the University of Kassel is a PhD student which used this program, and now we are trying to help colleagues from Ukraine to use the program for rabbits. And thank you for your attention.
Thank you, Emil. It was very nice lecture.
Yeah.
And very informative about how many research you do with your colleagues, and maybe more. Can you tell us more about EcoWeight?
No.
But EcoWeight is very useful, no?
Yes, we have a lot of users from, let's say, not only from Europe. We have users from USA, we have users in Africa, in Asia also. And as I mentioned, the new traits are incorporated by the user ask. For example, colleagues from Finland ask us to incorporate some traits. I don't know what is it. Forgot. No? Yeah. And for example, colleagues from Italy need the traits for cheese, and there's at minimum, let's say, this complex program costs we used minimum 500 inputs per one computation without the gene flow. You know that we are using a sometimes separate program for gene flow. Sometimes it is incorporated in the program. It depends on the complexity of the program.
So the output of the program are economic weights.
Not only, there are a lot of outputs. We divided to the three main parts. This is just outputs based on production and reproduction traits from statistics, and we have outputs based on economic weight, economic parameters. Very for a lot of categories, and the last part is economic output for raw, let's say raw economic weight, standardized economic weight, and relative economic weights. Of course, we use some genetic variances for standardized economic weights.
Yes, and if someone has something special, some special breed or a special, I don't know,
Yes, small
farms and
population.
so on, populations, you are able to, are you able to work on program in this way?
Yeah, we also already do it for some farmers. It wasn't a small one, but it was rather big farms. But it is possible to perform this program for optimization, economic optimization of the farms. Also.
So thank you very much.
Thank you.
Very interesting.
Yeah.
Yeah. So you have some question on Ariel also, maybe. Have anyone has a question? So thank you very much, Anel, and
I start sure for this
yes,
session. Yeah,
this session.
yeah, we have lunch.
We have lunch. Yes. Everybody who is not employed in this institute will have a lunch and the second part of this room around the noon, and parking. Sausages, but not from pork, but sausages from Berlin.
Yeah. No. To bylo ten víceméně nepoužitelný. Raz, 20 nahrává. Můžu vám to dát do kapsy zatím třeba. No to aby nevyšlo. So, dear colleagues, we will continue with last lecture, last presentation for today, our meeting. And I think I have to introduce our colleague, Lida, floor is yours.
Thank you. I will shortly speak about our department and our new project and our new experiment here on experimental farm in Czechia. This name of this presentation is mining data from modern technologies, including infrared spectroscopy. It's a really technological project, but I will speak about genetic. Department of Genetic and Breeding of Farm Animals is an integral part of Institute of Animal Science in Prague for a long time, for a very long time, maybe 50 years. And we now deal only with dairy cattle, beef cattle, pig, sheep, and horse, especially with dairy cattle, beef cattle, pig, some sheep we are trying, and some horse we are also trying. Poultry we didn't do for a long time. And what we use? We use the method of population genetics and molecular genetics together. For example, there is a single-step genomic blood method, and this method is used for estimation, for prediction breeding value, genomic breeding value in routine for many breeds in Czech Republic. For example, for Holstein breeds, now for some beef cattle breeds and so on. And there is a history with this method in Czech Republic and our department, because it's about 15 years ago, Professor Přidil, our senior scientist, started with this method, single-step genomic blood in Czech Republic. Yes, and now we are one of the first countries that started this the prediction in. So also we use the bioeconomic models and the bioeconomic weights, as my colleague Emil KRUPA spoke about, and this research is focused to do some selection indexes for breeds and for some livestock. And we work in production traits, reproduction traits, and any traits that are recorded during routine recording or monitored in routine. But also we are trying to find new traits for future breeding, like health traits, and now feed deficiency or methane emission. And there is about 3 slides about beef cattle breeding in Czech Republic, because we have really good and effective cooperation with Czech Breeders Association. My colleague Zdenka Veselá, this technician, is main researcher for beef cattle in Czech Republic. So this association was established in 1990. Performance recording was certified by ICAR, and in Czech Republic is about 25 beef breeds. So, and the Institute of Animal Science developed method for breeding value estimation or prediction for beef cattle in Czech Republic. And in the routine, there is a list of traits in routine. For these traits are predicted breeding values. It's calving geese grow of beef bulls at performance station, linear type traits, scrotal circumferences, and so on. Maternal fertility and what is important as very new phenotype is live under ultrasound measures for Aberdeen and Angus breed. For this trait, genomic breeding value are predicted since 2023. And the new traits in research are live animal measures for Aberdeen Angus, temperament of young animals since 2018, food and black spotting, and some traits monitored by breeders 48 hours after calving. It's other and peace scoring, temperament of dens, and newborn calf vitality. So now I will speak about our new research project, New Tech Diary. It's this really technological dairy cow. It's project funded by Agriculture Ministry of Agriculture Grant Agency. And there are 3 collaborating institutes, Czech institutes, Veterinary Research Institute in Brno, Institute of Animal Science in Prague, Czech Moravian Breeders Corporation. So you saw Petr FLEISCHER, this is a colleague from Veterinary Research Institute, me and other colleagues like Eva Kašna and so on, from Institute of Animal Science, and Zuzana Krupová from Institute of Animal Science. We take a part in this project, and also there are a lot of colleagues here from Czech Moravian Breeders Corporation. They are part of this project also. So we would like to develop the selection indices for breeding healthy and fertile cattle. And we would like to use some information about genetics, nutrition, physiology, and so on for this. So we need some modern technologies like automatic milking systems and so on, daily milk production, daily milk contents. And also what we need as new technology is milk mid-infrared spectroscopy of milk infrared spectroscopy. So we plan to do genomic evaluation, yeah? And some collection of health data, maybe digital, yeah? And then we would like to define new traits, new phenotype. So we started together with Department of Nutrition of this institute with colleague Dr. Miroslav Joch. We started feed efficiency experiment. We have experimental table with feed rate stroke by Farm for monitoring of methane. We use green feed, and we plan to monitor the cows for 6 weeks. And what we will measure: feed intake, methane production, milk yield, milk quality, rumination, eating behavior, diet digestibility, and the dietary nitrogen utilization using spot sampling of urine. Yeah? There is some pictures from our table. Yeah? there is a scheme of greenfield, greenfield. Greenfield is something like sniffer, but on the higher level, and machine that monitor gas fluxes like methane and nitrogen. So, and objectives from nutrition aspects are that the rapeseed meal will be replaced with calcium nitrite, and it will affect methane production that will decrease probably, and the digestibility, milk production, and feed efficiency that will increase. So it is because the reduced methane by lower methanogenesis due to reduction of nitrates to nitrates, leave more energy available for milk production, and this will lead to a higher increase in milk yield. From a genetic point of view, we would like to first establish a new phenotype. This new phenotype is predicted feed efficiency on the base of milk spectroscopy. Yeah? So first of all, we need some phenotypes, direct measured phenotypes for dry matter intake. Yeah? Some methane emission of cows in experiment, and also with infrared spectroscopy of milk for those cows, these cows. There is some scheme how gas from dry matter intake measured in experiment directly to some breeding value from feed efficiency. First of all, you must have some experiment with cows and measure this dry matter intake, milk production, body weight, milk spectral analysis, and so on. Then you can use some mathematical method like partial least squares or some artificial network and so on. And you will establish some prediction equation. And this prediction equation we can use for cows, for when we don't know the direct measure of dry matter intakes, but we can predict based on spectral data of milk their phenotypes for feed efficiency. So you have some prediction of phenotypes, not directly measured, and on this prediction of phenotype, you can use some blood prediction, probably genomic, and you will have breeding value from this predicted phenotype. So this is our objectives and what we're trying to do. And now something about methane. Yes, methane as the second most important green gas. So it is about methane production. 40% of methane is produced by natural processes, and 60% of methane is produced by human activity. And if we look more closely on this human activity, we can see that there is some extraction, storage and transport of fossil fuels. That is 35% of all methane of human activity. But and 20% is created by waste management and agriculture. Agriculture creates about 40% of methane. 40% of methane that is developed by human activity. And in Czech Republic, my colleague Eva Kašna had a look on this site, Fakta o klimatu CZ, and she found in year 2022, total of 119 megatons carbon dioxide equivalent was produced in Czech Republic. Yeah? Agriculture was responsible for 9.6 megatons, and it means that agriculture contributed 80% to total emission greenhouse gases created in Czech Republic in 2022. And what about cows? What about cows? So cows contributed to methane by 3%. And why? Because this 80% emission in Czech Republic was can be divided into 32% of methane, 32 nitrous oxide, and 16 carbon dioxide. 16% of carbon dioxide. Yes, it's divided. And this methane is from 91% created by enteric fermentation of ruminants. Yeah? And it was 3.6 megatons carbon dioxide equivalents, and it is 7% of total production of greenhouse gases in Czech Republic in 2020. So there is last slide about methane in Czech Republic, and thank you very much for your attention.
Thank you very much for a very interesting lecture. I open a discussion. Let's some questions. You are open. Or we can just open the general discussions, and let's do it like Luca already done. I lock the door and everybody have to answer one question. I already answered.
So thank you very much for that. You will be here. Sorry,
yeah.
I cannot speak now.
Okay. We are pleasure to stay with us in Friday afternoon. Yeah, I know. Nice weather. And I think it was very important to prepare, organize the activities like this. We need it because we understood this cost project like one of the biggest opportunity to prepare consortium for European scientific projects. You know, we need a collaboration. We have to stay up to date with at minimum with Europe, also with scientific world, and we not have to separate because the population, let's say, only population of dairy cattle are general. Let's say you know, we are using a few, let's say, artificial insemination tools. If you look behind to the history. So this is, I think this is a general problem and we have to solve together. So we are glad to organize this. And again, thank to Lida to organize this activity. So questions, lock the door. Oh, thank you.
And maybe if you want to stay for 5-10 minutes, we can sit, eat, and discuss some
Don't panic, make a
general question, research question.
Yeah.
Yes, and so share experience. If you don't mind, we can lock the door.
Yes, we have a lot of food here. Have to eat it, of course.
So
You
thank
know.
you very much.
So enjoy stay in Prague for our colleagues not from Czech Republic and have a nice trip home, hopefully. And everybody else enjoy the time.
Thank you.
Yeah,
Bye.
weekend. Thank you.